Awesome. Okay, so folks, welcome back or welcome to our first Pop Bio on the Dark Sides event of the year. It is the first, right? Yeah. Um, Featuring Dr. Deepa Agashe, who is an associate professor at the National Center for Biological Sciences in Bangalore, India. Deepa works on a wide variety of topics concerning broadly the genomics, ecology, and evolutionary biology of adaptation to new environments. I can tell you, as a microbial ecologist, some of the stuff that I've worked on has been quite deeply influenced by her work on codon bias and sort of non-synonymous and synonymous mutations in microbial genomes and how that affects things like gene expression. But I don't think that will be the subject of her talk today. Um, and we have a beautiful picture of a flower beetle here, which she'll be talking about in her presentation called Founders, Shapers, Colonizers Predict Trait and Population Dynamics in New Habitats. So we're all looking forward to hearing what you have to say, Deepa, and you can begin whenever you're ready.
Thank you. Thanks a lot for this nice introduction, David. It's really nice to see some names that I'm familiar with, whose work I've seen over the years, and maybe some faces eventually in the discussion. Like David said, I'm going to tell you today about some work with flower beetles that we've been doing in the lab for the past many years. And I'm going to focus today on how we can predict what happens to populations by knowing something about who are the colonizers, who are the founders of these populations, especially in the context of new environments as they are being colonized. So I'm going to start off with something very broad that everybody's familiar with. I think all of us sort of understand and agree with a lot of data over the last many decades that who is colonizing and how many of them are colonizing really has an important influence on how populations evolve over time. And I think a human migration history and story is a really fascinating example of this. As we migrated out of Africa and our ancestors colonized different parts of the world at different points of time, how many of us were there when we got there? Who else was there? Et cetera, et cetera. All of this has shaped our phenotypes, our traits, our history a whole lot. And so, in general, in evolutionary biology and ecology, people have been focusing a lot on specific kinds of aspects of these colonizing or founding individuals. And a lot of work has gone into asking what is it about the diversity of the colonizers? Why does it matter? Does it matter? How much does it matter? And when it matters? Similarly, partly because of reasons of diversity and partly because of demographic effects, we know that how many individuals colonize is also important. I'll just give you an example of the genetic diversity effects based on my work from many years ago in my PhD. So here I did experiments with the red flower beetle. It's a global pest species that's found across the world, eats flowers of different types, so wheat flower, other cereal flowers. And wheat is its preferred resource habitat. It lives in wheat, it poops in it, it mates in it, it eats it. So it's the whole world is wheat, right? And so I took four different strains of these flower beetles from the USDA stock center, and I either mixed them in one or two or three or four strains mixed together. So I created populations with different genetic diversity. So you see that on the x-axis here. And I set up these experimental lines in either wheat alone, which is the great resource that they're all happy with, or in corn, which is a really poor and suboptimal resource for them, or in a heterogeneous environment where there was one patch of wheat and one patch of corn, and they could move freely across. And I asked what happens to these populations over a number of generations. What I saw is that the extinction rates decreased a lot as genetic variation in the founders increased. The populations were more stable, so the coefficient of variation is also reducing as diversity is increasing, and the average population size is also higher. So this is just one of many examples from across the world showing that genetic diversity in the founders is sort of a good thing in general. Like I said, this is not surprising at this point at all. But a lot of other kinds of traits or characteristics of founders can also be important. And this is relatively poorly studied for population dynamics, and understanding longer term evolution. And that's going to be the focus of today's talk. So here's one example of the kinds of aspects of founders that can also matter. This is work from Brinda, who was a PhD student in my lab some years ago. Vrinda set up these flower beetle small short-term experiments where she allowed flower beetles to choose between either a wheat patch or a finger millet patch. So finger millet is dark brown in this cartoon here. And finger millet is a resource that's found very commonly in India. It's used by people as food, but it is very rarely infected or infested by these beetles, and they don't do so well on it. It's not as bad as corn, but it's not as good as wheat. So if you look at females that are young and that are isolated at low density as soon as they close, and you ask what is their fecundity, you'd expect that this is the best a female can have, right? She's alone, there's nobody to bother her, there's nobody to compete, and she is young. And so her fecundity is very high on average. If you now have these females age over time, again in isolation, the fecundity reduces. This is not surprising. This is commonly observed in most organisms. But then what's interesting is if you expose some of these females, even when they're young, to high density, that's the yellow box plot that you see here, you see that their fecundity also drops a lot, and in some cases even lower than what aging does to female fecundity. In general, what you find is with different combinations of age and crowding or age and high density that you expose these females to after closing, you find that fecundity is generally dropping. So there seems to be a cost in some sense that the females are paying in terms of their fecundity, either because of aging or because of crowding. What Rinda also measured here is how much is the preference for finger millet, which is the new resource being offered to these females. So she asked how many eggs were laid in finger millet. That's the y-axis here in the second plot. And what you see is that that also generally declines with age and with crowding, with the exception of these old high-density females, which seem to again have a higher preference for this new resource. So in general, we see this pattern of niche contraction, where as females are getting older or as they are experiencing crowding and presumably some stress associated with it, they are becoming more conservative and they're laying most of their eggs or all of their eggs in wheat, which is their preferred resource, even if they are experiencing crowding earlier so that they should be expanding their niche. We don't see that at all in these females. This is something we've seen in previous experiments as well. So in general, then we're finding that there's this decline in fecundity and in resource preference for a new resource when females are aging and when they are experiencing crowding. The only exception, like I said, is these old HD females here in blue. And we wanted to understand what's going on there. So we took some of these eggs in a separate experiment from such females and asked more about what those eggs are doing. What we're finding is that the average egg size is larger. So this is the this mustard old HD box plot you see here. So these females that are older and have experienced high density are laying larger eggs, and these eggs also have more lipid content per egg compared to the other contexts that I just showed you. So what we think is going on is these older high density females are potentially, this is a case of terminal investment perhaps, where as they're coming close to the end of their reproductive lifespan, they are investing everything they have into providing their eggs with a little bit more nutrition, and that allows these eggs to have a higher survival and a higher rate of development, or time of development is shorter compared to the other females that they have who have a similar background or different backgrounds coming in. So the question I want to focus on in today's talk primarily then is, do these kinds of effects, which we see manifesting, again, there's a huge literature on similar effects in many, many organisms. Do we see these kinds of effects having any role in longer term evolution? And so that's what we did next. So this is a work from a different PhD student in my lab, Shubha, who's finishing up her PhD now. And what she did is she set up different replicate populations of these flower beetles, again in a wheat finger millet patch environment, but allowed them to evolve for many, many generations over time. And she set up all of these populations with either young females that had experienced low density or young females experiencing high density. So they came from the stock population in which they were growing up. And the low density individuals, Shubha picked out as pupae and isolated them so that they grew up in the same environment in wheat, but they experienced either low density as young or low density until they were older individuals. And then they got to start and colonize these new habitats. So what's important about this experimental design is that we are trying to maintain the same genetic diversity in the starting populations. So we collect 100 individuals from each of these contexts, so 100 adults, and the sex ratio is generally 1:1, and so we have basically 50 adults of each sex in each population, and they all came from the exact same stock boxes. So diversity is the same and demography is the same at the beginning, right? So we're trying to rule out all of those effects so that we can focus only on the effect of the founder context. So there are 100 individuals that start every population from the same stock boxes, but those 100 individuals either experienced high or low density after eclosion into adults, and they were either young or old. What I mean by young here is 2 weeks of age, so after pupation, and then old is 6 weeks of age, which is getting closer to the end of their reproductive lifespan. They live much longer than that, but their reproduction drops a lot after 6 weeks. So what then Shubha did is every generation, she took 100 adults that emerged from the new generation, allowed them to lay eggs, a week later removed the adults, and then again the rest of the offspring get to develop without any adults around. So every generation, she's trying to maintain about 100 adults. What do we find? So here's population size over time on the x-axis. And the thin lines are showing you what's happening with each replicate line. There are 6 replicates for each of these founder treatments. And the solid lines here are showing you what's happening as the median of all the independently evolving replicates for that treatment. Unfortunately, after generation 10 or so, there was this parasite, likely Paranosema, that swept through all of our populations uh, and infected pretty much everything in the lab. And so it took us a few generations to get rid of it. And so this gray area is sort of highlighting the time when this infection was around and was observed in most of our populations in this period. Not every population showed signs of infection in every generation, but all populations did get infected at least for 1 or 2 generations in this time frame. So I'm just going to block that time off saying something funny has happened there. Of course, it's interesting from a selection point of view because there's an interesting new selection pressure that's being imposed, one that we didn't want, but it was there. And so we're trying to make the best of it. Okay, but we can look at what happens in the first 9 generations before this infection shows up. And what you find is that all these populations are bouncing up and down. This is partly because of negative density dependence and partly a feature of what I'm going to show you next, which is how these females are laying eggs, how many eggs are being laid, and where they're being laid. So if you analyze these trajectories in general, we find that the context of the founders does not have a role to play in explaining any of these fluctuations per se. It doesn't have a role in explaining population size on average, the harmonic mean, the geometric mean, whatever else, the CV of the populations, and so on and so forth, right? The only time that it does matter, the context matters, is in case of the harmonic population size, but that's if you also include these parasite infestation generations in your data set. So that's what you see here. This is the mean population size across all 15 generations. And you see that the old LD context, founder context, those populations have substantially lower population size on average. But this effect disappears if you remove those post-infection generations. So until the first 9 generations, everybody's behaving more or less similarly. What is going on with this infection-based population size? Why are we seeing these results? So here are some data showing you what's going on. What we're seeing is that over different generations, I'm showing you a heat map of pupil mortality. So every time Shubha was censusing these populations, she was noticing how many pupae seem to have died and shriveled up, potentially because of infection. We can't be sure about this. And the heat map shows you darker colors show you higher mortality. So every row here is a replicate population, and here are the treatments. And the dotted line marks where the parasite infection started being noticed in general. And what you see is that after After the infection started, the mortality of the pupae substantially increases in all of these replicates. But the young LD founder context somehow allowed populations to have lower mortality than other populations. And so this is something that we don't quite understand why, and we're testing some hypotheses that I'll mention at the end. So we clearly see that even after 10 generations of, the founder context being established long, long ago, there is still an interesting and important effect of parasite resistance being higher in some populations than others, depending on who their great, 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 great parents were and what they experienced. This is very interesting and surprising to me. In general, we expected going into this experiment that the founder context effects would last for maybe 1 or 2 generations and then they would dim out, because I don't expect that these effects on fecundity and preference for different finger millet or wheat would play a role for such a long time. And you'll see that in some cases it does. So what happens to other traits that are of interest and importance? Let me start off by showing you a little bit of data on fecundity and preference for finger millet. I showed you already earlier that these founder contexts, those females themselves, have different fecundity and preference. And that's what you see for generation 0 here. So this should not be surprising given that you've just seen data from Brinda earlier. In general, the young females are laying more eggs, have higher fecundity at generation 0, and they are having a high preference for finger millet in these experiments. The older individuals, the older founders, on the other hand, have lower fecundity overall, and they have low preference for finger millet as well. And then if you look at what's happening in generation 1, which is the daughters of these females that were tested in these assays, you're seeing that they're doing exactly the opposite very often of what their mothers did. And so the patterns are quite different depending on the context. So you'd expect that here the founder context should have a big impact in generation 1 in terms of these traits. And you'll see later that we do see that. But I want to first show you these trajectories of what happens to these same traits over these 15 generations of evolution. Again, the gray bars are marking areas where we were noticing infection. What you see is in general, for both fecundity and preference for finger millet, all of these populations, even though they start off quite all over the place, they converge more or less to these dotted lines, where the trait values in generations by generation 15 are kind of the same, right? What I'm showing you here also, every dot represents the median value of the replicate lines. So in each generation, Shubha measured 3 replicate populations out of the 6 that we had, just because she couldn't measure all of them. Logistically, it was hard. So the data, of course, for each line are more messy, but this is what the overall pattern looks like. And so what we see is that the traits eventually converge, but the way in which they get there is different for different contexts, because of course the starting point of the context is also different. I want to also mention here that what's happening in generation 11 and 15 is also not quite clear to us. It's possible that some of that convergence in generation 15 is driven by the parasite, which may be forcing everybody to sort of have a certain preference, a certain fecundity, because anything else is simply not possible given that there is infection. And so we're not quite able to test this directly because unfortunately all our populations got infected. So we don't have uninfected populations that stayed pristine throughout to compare with. So we're a little bit stuck with not being able to tease apart in the last set of generations what's happening due to the parasite and what's happening as a function of context anyway. But what is clear is that the context does not influence the trait evolution later on in the presence of the parasite, because then the populations behave very, very similarly. Okay, so I want to now show you the outcome of asking, are there consequences of these founder contexts on these different traits? I've only shown you data for a few traits here. And what we find is that in general, the founder context does shape some traits across several generations. Let me walk you through this figure. On the y-axis is the effect size, which we're estimating through fitting simple ANOVAs to each of these traits that we've measured. So first you see data for fecundity. And for each generation, Shubha has data from several replicate lines. So she's asking, is there a significant effect size, shown in pink, for the effect of age on fecundity, right? And you see that in the first generation, yes, there is a strong effect. It's significant. But over time, it fades away and wanes, and there isn't a significant effect anymore. Similarly, the age by density context effect doesn't exist. Everything that we see in the beginning for fecundity is explained by age or density alone. Similarly, you can look at what happens for all these different traits that we measured. You see for survival that the context never mattered. Survival in these habitats is very, very similar in general and is not driven by context at all. But these traits like preference for finger millet, they show strong context effects in the early days and surprisingly also in later generations. So in generation 11 and 15, you see that how fast the offspring develop, what I'm calling development rate here, as well as the preference for finger millet, once in a while does show even stronger effects of founder context than what we were seeing in the first couple generations. This is a bit surprising, and I'm not entirely sure what's happening here. We have a few hypotheses. One of the hypotheses we had was that this is mediated through diet. So remember, this is a new diet, finger millet, that's being provided to the offspring. And we know already that the founder context sets up a different preference for where the females are going to lay their eggs. We've shown in other experiments earlier that if larvae grow up being exposed to a new environment like finger millet, they're more likely to choose it for oviposition later on as adults. And so we thought maybe this is why there's this feed forward loop that's being set up. Early days, founder context influences preference for finger millet. That strong effect percolates across generations because in every generation, the larvae, if they're more likely to eat finger millet because their mom put them in the finger millet patch as eggs, then maybe that preference goes into the next generation and that cycle propagates. And perhaps this feed forward loop could lead to an increased effect size like what we see here. But when we measured stable carbon isotope ratios of what these individual beetles were actually eating over time, we don't see a founder context effect at all. And so I'm just going to summarize these data in this plot. The way we measured diet of these beetles is at every generation, Shubha was storing beetles in the freezer, and we went back to some of these stored samples for the first 6 generations, pulled out 5 beetles from each population, and sent those for analysis for stable isotopes. So because finger millet and wheat have different stable carbon isotope ratios, because one is a C3 plant and one is a C4 plant, we can measure the beetle in the beetle body, what is the percent finger millet that was being consumed. It's an approximation because there are several other issues involved with metabolism that make things difficult, but it's a good estimate. And what you're seeing here is on the y-axis, the percent finger millet consumed. And you can see that the different colors, which indicate the different treatments, the founder context treatments, there's no pattern there at all. But what is correlated with, what it does correlate with is the population size at that generation. So remember that even though we were setting up every population with about 100 individuals at each generation, because those 100 individuals themselves came from a crowded or an uncrowded context, or they were laying eggs and their offspring that were being produced were different. And what that meant is that the actual population size or degree of crowding that every population experienced was different. And this is a much better explainer of how much finger millet was being consumed by individuals in a given population at a given time point than the founder context or anything else that we've measured so far. So this feed forward loop, this nice idea that I had when we first saw these data falls flat because the preference doesn't correlate at all with the context of the founders. And so we're still left with a bit of a question saying, okay, well, how are these longer term effects mediated? The most likely explanation is that it's mediated through initial population cycles that are set up differently at a slightly different phase in some sense by the founders being either young or old or high density or low density from the context in which they came. This is something we're still figuring out. So I'll just wrap up this part of my talk. I've shown you that prior experience of colonizing individuals can shape trait evolution and sometimes far into the future, even though we don't fully understand it yet. We are sure, though, that these effects are not mediated by diversity and not immediately, at least by demography, because the population cycling in general is very similar. The patterns are similar. But it's possible that they are slightly asynchronous, and the amplitude of that cycling at critical points of time is set up by the founders. So because we're measuring our traits at fixed generations in every population's lifetime, it's possible that at generation six, the different populations are in different phases of how the founder context has set up things so that these great, 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 great grandchildren have produced slightly fewer offspring or more offspring, and so on. So we're going to try and tease these apart in with some new analyses in the next few days, months, and I'd be very happy to hear any other ideas or suggestions that people have since this work is something that we're wrapping up now. I want to move next now to what happens to longer term timescales. So, are there longer term effects of founders? And in this context, which traits actually matter? So, what I've shown you here is context of the founders mattering because they came from very, very different experiences themselves. But what about specific aspects of founders, like the traits that they carry? Which traits actually matter? So to answer this question, about 14 years ago, when I first set up my lab, we sourced populations of flower beetles from different parts of India, and we collected a bunch of wild beetles. Wild, quote unquote, here is just grain warehouses where grain is stored, and there are beetle infestations in many of these warehouses because these beetles are also super resistant to everything that you want, that you throw at them to kill them. So we collected a bunch of these beetles from different parts of India, and we brought them into the lab and set up large stock populations in wheat, maintaining as much of the diversity that we found initially in these populations. And then we set up replicate lines from each of these 10 source populations in corn. As I mentioned to you before, corn is a really bad habitat for them. Their survival is low, their development is slow, and just generally things are quite bad. And then we evolve these lines in continuous generation cycles over time. And we did continuous generations here because it's very tedious to do discrete generations for very long. And so these data that I'm going to show you now are data spanning 5 years of this experimental evolution. And the first thing we wanted to do was to make sure that these source populations, the ancestors of each of these replicate lines, that they have some differences to begin with, right? Because if they're all identical in their ancestral traits, then we don't expect to see any effect of what source population you came from. So here's just 4 of the traits that we measured for these ancestors. We measured how long they lived, so their lifespan. We asked how long they resist starvation, which surprisingly is up to 10 days without any food as adults. How cannibalistic are the larvae? Because this is shown to be beneficial for these in poor habitats. And how fast do they develop in corn? So the faster you develop in general, the better off you are because the faster you get to a reproductive stage. And so what you see is that there is variation across all these different populations in all of these traits that we think should be important for fitness in this new habitat. And the next question was to say, okay, what happens to these populations, right? So I'm showing you data here for the log of adult population size over time. You see this classic U-shaped curve here of evolutionary rescue, where the populations initially crash and they almost go extinct. But all of them, all 30 of them, except one, managed to survive, right? Avoided extinction. And so there's this beautiful rescue dynamic that we see. And then after that, they go on cycling up and down, primarily through density dependence, is my feeling, but we haven't specifically tested that. So what you see is that these dynamics overall are quite parallel. Remember, these lines all started with ancestors which had very different phenotypic trait values, as I just showed you. So the first thing we wanted to ask then is ask whether these details of this rescue event, which looks very parallel here, but which actually varies quite a bit across populations, do these details have some explanation in the ancestral trait values that I showed you for each population? So to ask this, we split up these initial 30 to 40 generations of data into these kinds of split regression models. So we first ask, okay, what is the slope of that first decline? How long does that bottleneck last? When does that first recovery take place? What's the slope of that, et cetera, et cetera. And the goal here was to say, if we fit these similar kinds of split regressions to each replicate line, because we have data for each line, here you see just all the data for all populations combined, just to show you what happens with these fits. But we can fit this for every line. And then we can ask, okay, we know the ancestral trait values for each of these lineages. Is there a correlation there at all? Can we see that the ancestral trait X is driving what's happening to this sort of rescue dynamic? So what I show you here is a heat map showing you the strength of correlations between each of these mean and variance in trait values for the ancestors, and each of these stages or demographic events in this rescue dynamic that I showed you before. And the upshot of it, only significant correlations are being shown. And the upshot is there isn't something that really sort of is consistent and strong in general. So there's this one correlation, for example, that's positive between total offspring and the start of this recovery. So populations that were started with founders who happen to have more offspring produced in corn in the early days, just right here, somehow have faster or faster recovery in this part of that rescue. And that's it. There's nothing else that's really dramatic or making sense. It's also surprising that no ancestral traits correlate with how rapid this decline is, how long this bottleneck lasts, how low is that bottleneck, and so on, right? So in general, we find that ancestral traits do not predict demography during rescue. And so this remains something we don't understand well. But if you look at what happens to the population dynamics over the long term, so across the entire 60 generations, if we bin these data into 10 generation bins just for simplicity and ask what is the average population size in each of these 10 generation bins, and now do the ancestors explain any of these traits, any of these patterns, we do see a nice and strong correlation across the time. So for example, here with development rate, which is how fast the offspring develop, both the average and the variance in this trait in the founders over here is explaining consistently what's happening to population size of both adults and larvae over the entire 70 generations of evolution. Remember that they were just started up like this with higher development rate or faster development in corn, and that is positively correlated with adult population size for 70 generations. So really, really long-lasting effects of who were your ancestors and what trait values they carried, and specifically the trait of development of offspring in corn. So this was kind of very interesting to see. And so then we expected that populations should be under selection to develop faster, given the correlation I just showed you. So we asked whether the development rate had indeed evolved over time. I'm showing you here data first for two of these populations. The green or blue is ancestral trait value. The generation one founders are in pink. And then in yellow are these three replicates measured at generation 70. And what you see is that in general, across all these populations, the development rate of offspring has increased substantially in most of these cases. And so, as predicted by our data, the correlational data, development of offspring is indeed something that's under very strong selection in corn. And this has evolved in general, showing signs of positive selection in all of our populations. So, what do we conclude? I hope that I've convinced you that the phenotypes of colonizing individuals can shape evolution far into the future, but the variation in the rescue parameters remains a mystery. So you might be thinking, okay, but maybe if bottleneck size didn't vary too much across the populations, there is no variation to explain. It did vary a lot. There's a fourfold, 11-fold variation in the different traits that I showed you for rescue. And we simply cannot, we don't have at the moment any idea of what determines exactly those details of how fast the rescue will occur. In general, we find that population dynamics converge over time. And overall, from both parts of my talk, I hope you've seen that details about founders can help predict population fate with some applications for conservation and pest control. I want to just quickly say that our long-term beetle lines continue in the lab. So I showed you data for corn. This is in yellow, up to about 60, 70 generations. We now have about 100, 150 generations of data for not only corn, but wheat control and finger millet selection lines as well. And so I'm really excited and interested in working with these data more to see what we find, what's the genetic basis of adaptation, what's happening with development rate in these other populations, how long does the ancestral trait variation effect last in general. So we continue to work with these lines, and I'd be happy and grateful to hear any ideas you have about what could be going on here. I'll end by thanking everybody who's been without whom this work would not have happened. So Vrinda here is the PhD student who spearheaded all of the analyses I showed you for the long-term evolution lines. But as you can imagine, these are data collected over 10 years and have taken enormous effort and time from many, many people. Uh, so Arun, Manju, Sham, and Shivansh helped with maintaining these selection lines over the years. Gaurav helped with some of the fitness data that I showed you earlier on the oviposition preferences. Shubha is a current PhD student in the lab who's finishing up, and she was helped by Draupadi and Pratibha in analyzing all of the data that I showed you for the context dependence and those selection lines. I'm grateful to NCBS for funding and support, and I'm happy to get questions and discuss. Thank you.